This section traces the two-century argument behind epigenetics. The question underneath it is single: can the environment write into inheritance? Lamarck said yes. Need shapes the organ, use transmits it, a soft inheritance in which what the body lives through passes to its offspring. Weismann built the wall against this. Germplasm separates from soma early, so nothing the body acquires ever reaches the gametes, and the modern synthesis hardened that separation. The chapter's thesis is that epigenetics reopens the case, the wall closed too soon. Waddington's canalization and genetic assimilation, introduced in 1942, show a heredity that is environment, responsive, and reversible, yet never alters the DNA sequence. It honors Weismann's barrier while restoring Lyman's barrier, while restoring Lamarck's channel. Its job in the book is to frame epigenetics as the delayed synthesis of a two-century dialectic, an old case reopened rather than a fresh discovery. The victor concedes that the loser was half right. This section explains why heredity runs on two channels rather than one. The first is the DNA sequence, the channel Weismann sealed off from experience. The second is a reversible layer of chromatin marks, cytosine methylation, and histone state that the environment can write and the germline can sometimes carry. The chapter's whole arc is a pendulum. Lamarck's soft inheritance is rejected by Weismann's germplasm barrier and by Mendel, then hardened into neo-Darwinism. It is resurrected once a mechanism appears, acquired change, transmitted without touching the sequence. Darwin's discarded gemmules return as small RNAs traveling from soma to gamete. The generative move is that a refuted hypothesis is readmitted the instant a mechanism licenses it. That move is what the chapter is for. Epigenetics names the missing channel. This section explains where the control over a gene actually sits. The genome is read only. Regulation lives one layer above it in how the fixed sequence is folded and where that fold sits in the nucleus. Packing 2 meters of DNA into a nucleus 5 micrometers across is more than a storage problem. The fold itself is the control. A gene's expression is set by two things: its conformation, whether nucleosome, fiber, or loop, and its address in nuclear space. Active clusters loop toward the interior. Silent ones press against the periphery and the lamina. Cells rewrite this geometry. ATP-driven remodelers slide and evict nucleosomes. Histone marks and methylation retune each unit's charge and stickiness. The chapter's move names chromatin as a dynamic access layer rather than passive packaging. The same substrate is held in an addressable state. This section explains why the folding of DNA is itself the regulator. 2 meters of DNA fold into a 5-micron nucleus, and the folding is not mere storage. It is the regulator. The generative primitive is this. Geometry gates access. Rack DNA on a histone octamer. Stack nucleosomes into a 30 nanometer fiber, loop and scaffold upward to the 700 nanometer mitotic arm. Each tier of compaction is simultaneously a tier of silencing, and every dial that loosens it, acetylation, methylation, ATP-driven remodeling, variant swaps, shear nuclear position, is a read and remodeling, variant swaps, shear nuclear position is a read and write permission on the same molecutic arm. Predict from this. Anything that repositions a locus toward the dense periphery should silence it, and toward the open interior should express it. The chapter is that one identity, packing state, equals expression state, elaborated. This section explains why chromatin regulation in plants works differently from animals. Animals commit their chromatin states early and then freeze them. plants refuse to, and that refusal is the entire claim of the chapter. A plant cell holds gene silencing as a live antagonistic equilibrium, never a settled switch. Two opposed machineries pull on it continuously. On the repressive side, PCG and PRC2 write the mark H3K27 Me3. On the activating side, TXG and ATX1 write H3K4 methylation. Underneath both, ATP-driven SWI SNF remodelers tune the physical access to the STP-driven SWI ESNA. The payoff of keeping this equilibrium unresolved is the meristem stem cell. It stays pluripotent yet instantly differentiable precisely because nothing was ever locked. Watch the gene WS in the shoot. WUS in the shoot, apical meristem. CAF1 restricts it. SYD sustains it, and the balance between them sets the stem cell number. You can now predict any plant case by asking which side of the repress to activate tug is being retensioned rather than which switch was thrown. This part lays out one structural primitive. Plant development keeps its epigenetic switches writable where animals weld theirs shut in the embryo. That asymmetry is the entire chapter. The primitive is chromatin state as a held latch. A latch holds the position you set it to and can be released and reset later, while a set, once fused, blows a single time and stays blown. Because a meristem must stay pluripotent, yet spawn differentiated progeny on demand, the same nucleosome hardware gets repurposed as reversible gates on master genes. That hardware includes assembly factors like CAF1, SWI, SNF remodelers, and PCG and TreeXG marks. CAF1 pins, WS to the organizing center. Cold latches FLC off for vernalization. Then each generation resets the latch. Read everything after as one move restated. Identity here is a maintained variable, so the machinery that maintains it is where control lives. This section explains how DNA methylation works as an epigenetic mechanism. Methylation is a covalent tag laid over the sequence, not written into it. A methyl group sits on a cytosine at CPG sites, one bit written on top of the code without altering a single base pair. Here is the generative move. CPG is a palindrome, so a fully methylated site becomes hemimethylated after replication, and a maintenance enzyme reads the old strand's mark to restore the mark on the new one. That single copy rule buys heritable memory the genotype never encodes. Identical DNA, two stable expression states transmitted through mitosis. Riggs and Holliday and Pugh proposed it precisely because nothing changed at the DNA level, yet the state persisted. Once you hold that, X in activation, and IGF2, H19 imprinting, predict themselves. Same sequence, with methylation deciding which copy speaks. This section explains the oldest regulatory layer alive, a single chemical tag laid over DNA rather than written into it. One covalent move, a methyl group parked on a base, cytosine or adenine, leaves the sequence untouched, yet makes identical DNA mean different things. The mark is not information inside the sequence. It is a tag over it, read by proteins that recognize DNA by shape as much as by letters, so it shifts their binding without editing a single letters. The chapter's thread runs through this. The same annotation is the oldest regulatory layer alive, and its root function is self versus non-self discrimination. In bacteria, a methyl tag flags a strand as mine, and restriction enzymes cut only what lacks it, the invading phage. Everything downstream, replication timing, silencing, imprinting, is that one move repurposed. It is an addressable overlay on a genome whose text never changes. This section explains how histone marks regulate genes through two separate channels, charge and address. A histone tail is a writable strip. A mark is a triple, which residue, which chemistry, and how many groups. The meaning is carried by that triple, not by the chemistry alone. Acetylation acts directly. It neutralizes the tail's positive charge, loosening its grip on DNA. Pure mechanics. Methylation acts indirectly. It moves nothing. It only changes whom the tail recruits, a label for effector proteins. So the same methyl group activates at K4 and silences at K4, ancillaries. The chapter's thesis is that chromatin state works as a combinatorial code rather than a simple switch, position, chemistry, and degree. This code is written by one enzyme class, erased by a second, and read by a third. This section explains how one genome can run different programs through an erasable second layer. The same genome runs different programs because a second, erasable layer sits atop the sequence. That layer is covalent marks on the histones that DNA winds around. The nucleosome is the switch. Tighten the wrap and transcription factors cannot reach the promoter. Loosen it and they can. Two levers act. Acetylation neutralizes the histone's positive charge, directly weakening its grip on DNA. Methylation instead recruits reader proteins that open or close the region. Because the marks are combinatorial and fast reversible, one nucleotide string encodes many cell states, even both at once, the bivalent promoter held poised in stem cells. Epigenetic information lives in what is written onto the packaging, not in the letters. This section explains why organismal complexity does not track gene count. Gene count barely separates a human from a nematode. Organismal complexity does. The inver becomes protein. That carries the whole story is this. Complexity scales with the regulatory layer that wires the protein parts together. The size of the parts list barely moves between organisms. What moves is the regulation, and that regulatory layer is written in RNA that never becomes protein. The central dogma read non-coding DNA as filler, as cushion, as structural scaffold. Yet 98.8% of the human genome is non-protein coding, and nearly all of it is transcribed. The program was mistaken for packaging. Maddox's move was to see that what you were calling junk is the regulatory information whose combinatorics build a brain from the same-sized parts bin a worm uses. Every non-coding RNA class, the later chapter's catalog, is one instance of this single reframe. This section explains why the vast untranslated part of the genome is the regulatory apparatus itself, and why that inverts the central dogma. The central dogma reads RNA as a courier, DNA's message in transit to protein, and treats everything untranslated as noise or padding. This chapter inverts that. In a human, 98.8% of the genome is transcribed yet never translated, and that fraction is the regulatory apparatus itself. The step that forces the argument is Maddox's comparison of humans and nematodes. The protein coding gene counts are near identical, yet the organisms are wildly different. That comparison forces the conclusion that complexity scales with regulatory information rather than with part count. The generative primitive is this: an organism is specified by its wiring rather than by its inventory of parts. Non-coding RNA is where that wiring lives, and the rest of the book's mechanisms hang off it. This section explains why a single trick controls where epigenetic silencing happens. Specificity in epigenetic silencing is factored out of the machinery and encoded as a short RNA sequence. Every effector here, whether a chromatin-modifying complex, an endonuclease, or a translational break, is generic. What tells it where to act is a non-coding RNA, whose complementarity to the target supplies the address. Base pairing is the addressing scheme. The chapter's two size classes are one protocol. Convenience cut across a single pattern. Long non-coding RNAs run from 200 nucleotides up, and the small ones sit at 20 to 30 nucleotides. MicroRNA, small interfering RNA, and PIWI interacting RNA differ only in how the guide is excised and loaded. Once you read silencing as a guide plus a generic effector, every kingdom-specific variant in the chapters that follow is a substitution in one slot. This part explains how a cell silences a gene, and where the surprise in that mechanism actually sits. Silencing a gene needs two separable things, an address and an actuator. The pattern across this chapter is that a non-coding RNA supplies the first, and a conserved protein complex supplies the second. A guide of 20 to 24 nucleotides finds its target by base pairing. Argonaut, or a chromatin modifier like PRC2, does the killing. Swap the guide, we target the same machine, and the genome becomes programmable, silencing, without editing a single coding G. This is why one RNA apparatus spans bacteria to human, why a single dicer serves thousands of microRNAs, why deregulation reads out as cancer. The surprise is where specificity lives. It lives in the cheap disposable RNA rather than in the expensive conserved protein. This is indirection, biology's version of it. The guide is a pointer. This section explains the prokaryotic small RNA as a single design principle. Regulation needs no protein when a folded RNA already is the recognition itself. The prokaryotic small RNA is a shape, and everything it does follows from whose shape it copies. Copy the target messenger RNA sequence, and you base pair to silence it. This is what the cis, encoded and trans-encoded small RNAs do, and what the CRISPR RNA, the CUR RNA, does against phage. Copy a protein's natural substrate instead, and you sequester the protein. 6S RNA mimics a melted promoter to trap RNA polymerase. CESRB mimics a messenger RNA. GLMI mimics another small RNA. Copy nothing, just fold a ligand pocket, and you sense a metabolite directly. This is the riboswitch. One primitive runs underneath all of it, recognition by complementary geometry. The intercepted target is the only free variable. Bacteria ran this before eukaryotes named the microRNA. This section explains how RNA can regulate without ever making a protein. It reduces to one move. A non-translated RNA presents a shape or a sequence that some existing machine already recognizes, and it competes for that recognition. Two flavors generate the whole chapter. In the first, the RNA mimics a protein's natural target and sequesters the protein as a decoy. CSRB looks like messenger RNA, 6S looks like melted open DNA, GMY looks like another small RNA. In the second, the RNA base pairs with the target messenger RNA directly, either cis-encoded or NA directly, either cis-encoded or trans-encoded, and it blocks the ribosome or recruits a nuclease. Here is the chapter's real payload for a book about eukaryotic epigenetics. Bacteria were doing this first. RNA1 was throttling plasmid replication in 1981, years before anyone found microRNAs. Recognition-based control is ancestral. It predates the eukaryotic lineage entirely. This section explains how the same small RNA machinery behaves differently across the simplest eukaryotes. The small RNA machinery that tunes gene expression in animals appears in the simplest eukaryotes, doing something more violent, editing the genome itself. A short RNA is only a sequence matching probe. What varies across life is what the sequence matching probe. In the probe, ciliates make this legible. During conjugation, bidirectional germline transcription yields double-stranded RNA, diced into scanning. RNAs of about 28 nucleotides called cnRNAs. These scanning RNAs compare the germline against the maternal soma and physically delete the roughly 95% that fails to match, transposons. Fungi run the milder version, silencing rather than excision. One primitive holds across the kingdoms. Hand a cell short. RNA queries against its own DNA, and the readout is free to escalate from degradation to outright deletion. This section explains one conserved molecular apparatus that recurs across every eukaryotic kingdom, from protozoa and fungi up through plants and animals. The apparatus dices double-stranded RNA into guides of roughly 20 to 30 nucleotides, loads them into an Argonaut protein, and lets base pairing homology act as an address. What that address points at is an address. In most lineages, the address points at messenger RNA. In ciliates, it points at the genome itself. Paramecium and Tetrahymena use small RNAs to physically delete DNA, comparing the parental nucleus against the developing nucleus to decide what to excise, and doing so heritably. Oxytricha goes further still. Its RNA templates even set chromosome. The chapter's job in the book is this. The silencing core is ancient and invariant. Its target register, whether transcript, chromatin, or outright genome surgery, is the swappable part. This part explains how 3 animal small RNA classes reduce to a single silencing machine. One protein does the silencing. A short RNA says what to silence, fact the machine into a generic Argonaut, effector plus an interchangeable address strand, and every animal small RNA class becomes the same object seen through a different feed. MicroRNA, small interfering RNA, and PIW interacting RNA differ only upstream which precursor loads the guide and which peripherals shape it. The first 2 load from double-stranded RNA. PIWI interacting RNA, PIWI interacting RNA loads from single-stranded RNA. The peripherals vary too, 1 dicer or 2, an RNA-dependent RNA polymerase amplifier present in the roundworm C. elegans or absent in fly and human, soma, or germline. This is the animal column of a 3-kingdom table. Given the factorization, you can predict the protista and plant columns before reading them. The class names label the input. The engine never changes. This section lays out how animal gene silencing is built from a small fixed alphabet, and how one chapter holds that alphabet steady while varying only the wiring across 3 organisms. 3 small RNA classes, microRNA, small interfering RNA, and PIWI interacting RNA, dice out of a shared protein set, dicer Argonaut and PIWI, and RNA-dependent RNA polymerase. Together they compose the whole animal silencing repertoire. The chapter's move is to hold that alphabet fixed and vary only the wiring across 3 model organisms, T. elegans, and the marry only the wiring across 3 model organisms, C. elegans, Drosophila, and human. Read one lineage's composition and you generate the others by substitution and deletion. Worms amplify a faint trigger through secondary small interfering RNAs built by RNA. Dependent RNA polymerase, turning it into systemic transitive silencing. Flies and mammals simply dropped that amplifier. The odd component is PIWI interacting RNA. It has a single-stranded precursor, it is restricted to the germline, and it guards the genome against transposons. It is the exception that exposes the two double-stranded derived classes as the default axis. Everything else varies around. This section explains the RNA silencing machine as it runs in plants. One silencing apparatus runs in every kingdom. It chops double-stranded RNA into guides 21 to 24. Nucleotides long, hands each guide to an Argonaut protein and lets the guide sequence find whatever transcript matches, then destroy it or methylate the DNA behind. Plants are that same machine after gene duplication became division of labor. Where an animal runs 1, Dicer, Arabidopsis runs 4. It also carries 10 Argonauts, 3 RNA-dependent RNA polymerases, and 2 further polymerases, and 2 further polymerases, Pol4 and Col5, that exist nowhere else and are built for nothing but silencing. Duplicate apart. Specialize each copy to one precursor and one size class, and a single apparatus fans out into parallel pathways. The RNA-dependent RNA polymerase copy is the TEL. It amplifies the guide, so silencing self propagates. The output is a mobile, heritable immune signal that travels between cells and passes down through generations. This section explains how one molecular operation, run again and again, produces every class of small RNA in plants. Everything in this chapter is one machine run many times. Take a double-stranded RNA and dice it into fragments about 21 nucleotides long. Load one strand into an Argonaut, then cleave whatever base pairs to it. That single fold, duplicated across the toolkit, generates every class of plant non-coding RNA. Plants took the shared eukaryotic core, dicer, Argonaut, and the RNA-dependent RNA polymerase that animals largely lack, and expanded it by paralog duplication. In Arabidopsis, that gives 4 dicer-like enzymes, 10 Argonauts, and 3 RNA-dependent RNA polymerases, all running the same operation on different substrates. The difference from animals that drives all of this is the RNA-dependent RNA polymerase. It rebuilds a second strand from a lone one, so silencing, amplifies, and pathways chain. A single microRNA cut fixes the phase for an entire downstream run of trans-acting small interfering RNAs. Predict a new class by asking which enzyme paralog dices which substrate. This section explains why plants and animals build the same tiny RNA guide through 2 differently factored pipelines. Both kingdoms make the same product, but they split the labor of one pipeline differently. Each must carve a guide of about 22 nucleotides out of a hairpin transcript, and doing so takes 2 cuts by RNA's 3 enzymes. The whole story is where those 2 cuts happen. Animals split the cuts across the boundary of the nucleus. Drosha, working with its partner, DGCR8, makes the first cut inside the nucleus. Dicer makes the second cut out in the cytoplasm, so the precursor microRNA crosses the nuclear pore mid-processing, half-finished. Plants collapse both cuts into a single enzyme, DCL1, acting entirely inside the nucleus. The finished duplex is then exported, methylated by HEN for protection on the way out. The downstream silencing logic tracks this split. Animal guides pair imperfectly with their targets and throttle translation. plant guides pair almost perfectly and cleave the target outright. The 1D point is this: where you place the compartment boundary fixes both the enzyme count and the silencing mode. Choose the boundary, and the rest of the pipeline follows. This part reduces RNA silencing to a single program. A short RNA of about 20 to 30 nucleotides is a programmable address. Base pairing names the target, and an Argonaut protein is the executor that acts on whatever the address points to. Everything the chapter tabulates, from myRNA through pRNA, plant versus animal, is parameter variation on that one scheme. The parameters are two. Where the guide gets cut out, plants finish entirely in the nucleus, while animals hand it off from the nucleus to the cytoplasm. And how tight the match must be. A near-perfect match cleaves the message, while a seed-only match must be. A near-perfect merely blocks its translation. Convergent function, divergent machinery. The two kingdoms evolved the same address and executor logic independently, so you can predict any new class by asking three things: what precursor, cut where, and matched how tightly. This section explains how a gene can go silent without any change to its sequence. A gene need not mutate to fall silent. It need only meet its own likeness. When two homologous sequences share a nucleus, the quieter one can impose its silence on the active one. The expression state copies sideways, sequence to sequence, while the DNA stays untouched. And the freshly silenced copy is now itself infectious, converting the next active. Copy is now itself infectious, converting the next acture. Paramutation, transactivation, transvection, and co-suppression are four names. For one move, homology is the channel. The genome reads sequence, identity as an address, and routes regulatory state along it. This routing is heritable, reversible, and orthogonal to Mendel, who only ever let information flow down the generations, never between partners, who only ever let informations never between partners in the same cell. This section explains paramutation and the wider class of genomic communication it belongs to. Two copies of the same sequence can sense each other and equilibrate their expression state, with no change in the DNA at all. The silent copy imposes silence on the active one. The convert inherits that silence, and then it goes on to convert the next partner it meets. That is paramutation. The chapter's move is to read it as one instance of a wider class, with transactivation, transvection, and co-suppression as its siblings. The unifying idea is that homology itself is a communication channel, and state flows down that channel towards silencing, both heritably and contagiously. Here is the violation that makes this matter. Mendel assumed that alleles pass through a cross informationally sealed from one another. Sameness of sequence is precisely the coupling that breaks that seal, giving inheritance of a phenotype that the genotype does not encode. This section explains bacterial adaptive immunity, the system called CRISPR, as a form of self-editing registry. Immunity works here as a registry. The cell edits into itself. The cell slices a fragment of the attacker's genome into its own locus, then transcribes that archive into small guide RNAs that match and destroy anything carrying the same sequence. Defense collapses to a single operation. Pattern match against stored signatures. The enemy's own code becomes the recognition key. The surprise that reorganizes everything is that this memory is heritable. It is an acquired defense written into DNA and handed to daughter cells. This is adaptive immunity with no immune system, Lamarckian inheritance made mechanical. In the book's arc, CRISPR is the proof that prokaryotic adaptation is epigenetic acquisition. foreign DNA is captured, indexed, and reused as the template for sequence-directed degradation. This section explains why a bacterial immune system belongs inside a book on epigenetics. The whole idea is this: a cell that survives an attack keeps a piece of the attacker and hands it to its children. Bacteria excise short fragments of invading phage, DNA, and file them in order between palindromic repeats. Transcribing that archive yields guide RNAs that recognize the next infection by sequence identity and destroy it. This is why CRISPR sits inside an epigenetics book. It is heritable, acquired, sequence-addressed memory, defense, written into the genome as a chronological ledger of what once tried to kill you. Predict the rest from the primitive. All the specificity lives in the stored sample, so a single point mutation in the phage slips past it. This section explains how a cell tells its own genetic messages apart from an invader's. It does so without ever recognizing shape, only sequence keyed to one structural tell: double-strandedness. Single-stranded RNA is ordinary traffic. A double-stranded copy is the signature of something replicating that should not be a virus, a transposon. RNA interference reads that double-stranded RNA as an alarm, dices it into short guides, and destroys every transcript matching the sequence. That is the whole primitive, self versus non-self decided by homology, not by structure. It is an immune faculty older than antibodies, running on base pairing. The corollary predicts itself. Force one of the cell's own genes into a double-stranded form, and it gets silenced as if it were foreign, which is exactly how petunia breeders stumbled onto it. This section explains how a cell tells self from foreign using nothing but the shape of nucleic acid. A cell that recognizes foreign by sequence alone needs no antibodies. Double-stranded RNA is itself the alarm, since only viruses and runaway transposons make long double-stranded RNA. Dice it, retain one strand as a template, destroy every transcript that matches. Complementarity becomes the self versus non-self discriminator. Here is the part that shifts your model. This defense was never narrowly immune. The identical machinery, once it existed, was exapted into chromatin control, expression tuning, cell identity, and development. One mechanism read two ways: an ancient response to foreign nucleic acids and a universal regulatory dial. From that, you predict the rest. Anything presenting as double-stranded RNA, viral or endogenous, is silenced by the same homology rule. The chapter merely walks the RNA interference and virus-induced gene silencing instances. This section explains how epigenetic inheritance survives across generations when the germline deliberately wipes the slate clean. Heredity across generations does not copy the parental state forward. It works by controlled erasure, followed by a rewrite. The germline drives two waves of demethylation that strip the parental epigenome almost bare, and then the enzymes, DNMT3A and 3L, reinstall methylation, de novo, building the marks fresh. This creates a paradox for epigenetic memory. Totipotency, the power to become any cell, demands a blank slate. Yet imprints and transposon silencing must persist. Both cannot be true of a stored record that simply survives. The resolution is persistence by reconstruction. What must be remembered is actively rebuilt each generation rather than carried through intact. The piRNAs hold the memory of which sequences are parasites, and they remethylate the transposons in the germline, silencing them anew. Memory here is a rederivation protocol rather than a stored copy. That is why germline tissues must be reminded to silence what they already silenced. This part defines heredity by function rather than by carrier. Heredity is any structure that both templates the next generation and survives the reset between them. The genome is the default carrier. The chapter's move is that methylation, histone marks, and small RNAs form a parallel one. Inheritance counts only where the mark escapes reprogramming, the erase and rewrite that normally wipes the slate each generation. The extreme anchor is the ciliate. In Oxytricha, a maternal RNA pool rebuilds and unscrambles the somatic genome, so acquired rearrangements pass on while the germline sequence stays untouched, copying without the DNA. Predict the rest from this. Whatever reliably reconstructs the offspring and dodges the reset inherits, sequence or not, even cortical geometry, carrying no DNA at all. This section explains how cancer silences a tumor suppressor gene and why that silencing can be read and, in principle, reversed. A tumor suppressor gene can fall silent in two ways. You can delete its code, or you can leave the code intact but unread. Knudson's two-hit theory does not distinguish between them. Both alleles must be inactivated, and a hypermethylated promoter counts just as fully as a mutation. That is the chapter's move. What carcinogenesis manipulates is expression, the reading of the gene. The methyl mark is a second hit that changes no letter of the sequence. The asymmetry this hides is what the whole reframing rests on. A mutation is hardware damage, largely irreversible. An epimutation is a settable switch, so removing the promoting stimulus can undo it. Cancer is thereby partly software. That is why every stage of the disease leaves a readable methylation signature, and why the silencing is, in principle, reversible. This section explains how cancer stops being a story about broken sequence. Knudson's two-hit rule is the primitive. A tumor suppressor dies only when both copies fail. The chapter's move is that a hit need not be a mutation. Hypermethylating a promoter silences the gene as completely, without altering one base. Same lesion, different substrate. This buys what sequence damage cannot. First, a paradoxical dual signature, genome-wide hypomethylation loosening repeats into instability, while local promoter hypermethylation switches suppressors off. Second, reversibility, since methylation is written and erased rather than carved. That reversibility is why the chapter turns clinical. The epigenetic hit is a biomarker you can stage and a target you can undo. This section explains how experience leaves a lasting mark on gene expression without changing the genome itself. The genome is fixed. What varies is a writable layer above it, methylation, histone marks, non-coding RNA. This layer sets which genes are readable without touching the underlying sequence. The chapter's single move is this. Experience writes to this layer, and the writing persists. Meany and Sif's rats carry the compression. Maternal licking in the first week of life demethylates the pup's glucocorticoid receptor promoter, fixing a lifelong stress set point. Cross-fostering reverses it, and it transmits across generations with no gene altered. Every disorder that follows is one template. Rett, Alzheimer's, Huntington's, schizophrenia, a broken writer or reader corrupts the mark layer and gene expression drifts. Behavior and pathology are the same phenomenon, perturbations of a mutable memory sitting over a static genome. See the layer. Predict each instance. This section explains how experience becomes heritable without touching the genetic sequence itself. The genome is read only. The epigenome is the layer experience can write to without touching a single base. The move in this chapter is that behavior itself becomes a heritable channel. Meany and Sif's rats make it concrete. a mother's licking demethylates the glucocorticoid receptor promoter in her pup's hippocampus, tuning its lifelong stress response. Cross-fostering rewrites that mark, so the inheritance rides on care rather than on sequence. The same silencing machinery the book tracks against viruses here inscribes maternal attention into a maternal attention into a nervous system. The boundary that reveals the structure is that the mark is patterned rather than uniform. Methylation peaks and valleys cluster at synaptic genes, so experience writes with an address rather than a broadcast. This section explains why the genes alone cannot determine an organism's traits and where the missing variation is written. The chapter turns on an objection from Craig Venter. There are too few genes to fix an organism deterministically. The coding sequence is nearly the same across species, so the diversity of phenotype has to be written somewhere with more degrees of freedom. That somewhere is the epigenome. The settable layer of methylation marks that gate which genes fire. Environment, meaning maternal diet, stress, pollutants, and radiation, writes on those marks while the sequence itself stays fixed, and phenotype follows from what gets written. Read the whole chapter as a single substitution. Replace the claim that genes cause traits with a two-step claim. Environment inscribes the epigenome, and the epigenome regulates the genes. The prenatal window is where those right permissions run widest. The Dutch hunger winter cohort carries a famine's methylation signature that is still legible decades later in the survivor's own DNA. This section explains why your genes cannot be the whole story of who you are and what fills the gap. Genes underdetermine the phenotype. Venter's count is the surprise, too few to hardwire a life. The gap is filled by the epigenome, the writable layer where environmental history, maternal diet, stress, air pollution, toxins, radiation is inscribed as DNA methylation and read back out as altered gene expression. Environment leads to epigenome. Epigenome leads to phenotype. This is one causal chain, and its middle term is the mechanism the chapter exists to name. The Dutch hunger winter cohort carries it, famine before birth, and the methylation of the IGF-2 gene still shifted 6 decades later. The prenatal window is where the ink stays wet. From this, you can generate every case. Any exposure reaching the methylation machinery leaves a phenotype without ever editing the sequence. This section explains hairpin-based anti-sensing, a way to silence a gene by feeding the cell's own antiviral defense, a decoy. Cells already run a machine that shreds double-stranded RNA, an ancient antiviral reflex, because in a healthy cell, only viruses and copying errors produce double-stranded RNA. Hairpin anti-sensing feeds that reflex a decoy. You encode a transgene whose transcript folds back on itself, a sense arm, a loop, and an antisense arm. The cell reads this self-made hairpin as though it were viral. Dicer dices it into small interfering RNAs, and RISC loads one strand as a search key and destroys every matching RNAs, and RISC loads one strand as a search key and destroys every matching messenger RNA. The surprising part is that you never touch the target gene. You write the sequence you want, gone into the arms, and let the genome's own defense perform the deletion. The specificity is programmed by base pairing alone, and the silencing works by mimicry, leaving every base of the gene itself unchanged. This section explains how a cell's own antiviral defense becomes an instrument you can program. A defense mechanism becomes a programmable instrument the moment you can forge its trigger. The cell's RNA interference machinery hunts double-stranded RNA because that shape signals a virus. It dices the intruder into small RNAs and destroys everything matching their sequence. The hairpin construct exploits this literally. A transgene folds into a self-complementary stem. The machinery cannot distinguish from viral RNA, so it processes your decoy and silences whatever gene your arms were written to match. That is the whole chapter and the book's closing turn. Every mechanism the earlier chapters described as ancient antiviral immunity is here reread as an addressing scheme. Sequence complementarity is the address, and you supply it.